heavy lift rockets and the autonomous rovers. We're even testing the self-deploying habitats meant to house the first generation of pioneers on the Martian surface. But there is a biological gap in these plans. If we cannot reproduce off-world, these multi-generational settlements are temporary outposts with a fixed expiration date. We already know space is hostile to adults. Extended microgravity leaches calcium from bones, atrophies the body's muscle, and interferes with how cells communicate. But we have no data on the most vulnerable stage of life. What happens to the microscopic blueprint of a human embryo when gravity is simply removed? Until we solve this bottleneck, the vision of humanity as a multi-planetary species remains strictly theoretical. Subjecting a developing nervous system to cosmic radiation and weightlessness is a practical and moral impossibility. We cannot simply send pregnant astronauts into orbit to see what happens. Because of this, space life science has traditionally relied on the mammalian cycle shown here, using mice and rats as proxies. These models reveal broad biological trends, but lack the specific milestones that define early human growth. They are close, but they aren't us. And then there is the legal barrier. International bioethical standards, such as the 14-day rule, place strict limits on how long human embryos can be cultivated in a lab, effectively blocking research into the early stages of our development in space. These two constraints, the danger to human life and the rigidity of international law, have kept scientists in the dark, unable to study our own origins beyond Earth. The Tiangong 10 mission to the Tiangong Space Station carried a specific payload meant to bypass these roadblocks. It delivered engineered human-derived models designed to bring the study of our reproductive limits into orbit. These are called blastoids. They are engineered from stem cells to mimic the architecture and molecular signals of a real embryo, forming this highly structured, hollow sphere. Crucially, blastoids lack the ability to develop into a full fetus. Because they cannot become a human baby, they exist in a legal category separate from the restrictions placed on real human embryos. This technical workaround provides a high-fidelity proxy for scientists to observe how our biological programming reacts to a weightless environment. The experiment focuses on a narrow window between days 14 and 21 of development, a period embryologists call the black box. This is the stage of perigastrulation. It's the moment a single layer of cells begins to organize, deciding top from bottom and head from tail, while forming the precursors for every major organ. On Earth, gravity has been a constant, invisible anchor for this process for billions of years. It provides the orientation cells use to build a structured body plan. Aboard Tiangong, an automated robotic system incubates and monitors these blastoids, maintaining their environment in complete microgravity for 5 days. This is the core test. If you remove that gravitational anchor at the exact second the biological blueprint is being drawn, does the body's code adapt, or does the organization fail? After 5 days, the blastoids are frozen in orbit, their cellular state perfectly preserved for the long trip home. Back on Earth, scientists will compare these samples against a control group grown under the exact same conditions, but with the steady pull of Earth's gravity. And if the space samples develop normally, it suggests that human life could be self-sustaining, even during the years-long transit to a distant world. But if the cells fail to orient themselves, it proves that gravity is an absolute requirement for human creation. The data from Tiangong will dictate the terms of our future. We will either be biologically cleared for the stars, or we will have to develop large-scale artificial gravity systems before we can ever truly leave our cradle.